as I mentioned earlier when we kicked off the conference, it's always been important to us to cover a wide range of topics, a wide range of applications and fields, because data science is very broad. And one aspect of data science, which has become sort of increasingly important and interesting during this year, is biostatistics in general and sort of applications related to the pandemic in particular. So we're super happy to have two speakers here today who are biostatisticians and a data analyst coming from academia. So we're covering that angle as well. And they are going to tell us about the COVID symptom study in Sweden, applications of biostatistics and data science in e-epidemiology. So I want to welcome Marlena Maziarz and Neli Tsereteli. They will be presenting jointly, and we're super happy to have you here today. Over to you.
Thank you so much. So let's see. And can you see my slides?
Yes.
Okay. Okay, thanks for the introduction and thanks for having us. So my background is in, just a short introduction about me. My background is in human biology and computer science. I did a PhD in biostatistics in Seattle. So after that, so I guess the human biology and computer science was in Toronto. Then I worked as a data analyst for a few years. So I guess that's important because that motivated me to do a PhD in biostatistics. So after my PhD in biostatistics, I did a postdoctoral fellowship at the National Cancer Institute. And since 2018, I'm at the Lund University Diabetes Center working as a biostatistician. So I will introduce the COVID symptom study very briefly. I will focus on the statistical aspects of the study, since those are the most interesting, at least to me. So I'll talk a little bit about the challenges. I'll discuss the methods that we're using to make predictions. I'll show you some of the results, and Neli will continue with some examples in data science that she has worked on, that she applied to this study. So the COVID symptom study was, it's an app. So we collected data using an app. The app was developed by a company, Zoe, in the UK. It was launched in the UK in March, March 24th last year, and in Sweden it was launched on April 29th. So quite early in the pandemic. So currently we have over 4.5 million participants worldwide, and over 200,000 of those are from Sweden. And that includes over 12 million assessments from Swedish participants. So we collect data on, so postal code so we need the location where the person lives, but then we collect information on health status, comorbidities, a lot of symptoms. I will go through some of them. COVID test results, both PCR, so both active infection and antibody tests, but for prediction we use only the PCR tests. Information on whether someone is a healthcare worker, and if they are a healthcare worker, whether they work with COVID patients or not. We also allow for free text comments, which actually Neli will go into that, so I won't talk about that anymore. But and since 2021, we also collect information on vaccinations. So the data is available to researchers at King's College, Lund University, Uppsala, and Harvard. And the aggregated data for Sweden is publicly available. So wait, why can't I move my slide now? Okay, so here's I start with this slide because I want to emphasize the fact that this is a collaborative effort. So we have 3 PIs, 2 biostatisticians, data analysts, data scientists, a clinician who's a postdoctoral fellow also, a PhD student, and our project coordinator. So the main goals of this study I will not go over all of them. I just I emphasize the first one because that's the one I will be discussing today. So the main goal that we are interested in today is to monitor temporal and geographical trends in the spread of symptomatic COVID. And there are several other, so the work on this study is ongoing, and I think it will be going on for a while because I mean we have so much interesting data. So so we have a lot of other goals as well. So in this slide, I want to show you kind of, so on the left is what my life looked like before the pandemic, and on the right is what my life looks like now. And so before the pandemic, as a biostatistician, I would usually, can you see my mouse also? I don't know if you can.
Yes, we can.
Okay, thanks. So as a biostatistician, normally I would get involved into the study or be involved into a study at this point. Ideally, it would be here or like at the study design stage, but usually it would be here. So typically one would start with a scientific question, have a study design, collect the data, do data cleaning, exploratory data analysis. This would be a cycle. Then you do some modeling, maybe cycle here a little bit, but probably not recommended. And you then go on to the results. And in the pandemic era, everything is turned upside down, including this whole I don't know what you call it, like a flow chart or a sequence of how I would normally do things, which makes it actually quite interesting. And so I'll discuss a little bit the aspects that are most interesting to me. So the main thing that I find challenging is this part, the data collection being dependent on continued voluntary contributions from Swedish residents, which means that we don't, our sample is very random, right? We don't actually know what our sample is. It's quite biased. There are all kinds of biases, and so some of them I want to go into. And so the main problem with main challenge is to account for these biases. So I also want to say that so the other thing that is needed is that we need information from other data sources to kind of figure out what are the biases. So typically you will be able to say, okay, okay, you know what, I don't have time actually to go into the details, but I just want to say that using external data, we are able to see or try to infer what kinds of biases we have in our data set. So some examples of the biases that are the main ones. So we notice that participants joining the study are much more likely to be symptomatic at the time they join. Some participants report having symptoms for a long time after testing positive, so long COVID. Many more women than men report. Our participants tend to be younger than the general population, and the proportions across the regions also vary. So one of the main fixes is the inverse probability weighting, which I will discuss in more detail in a minute. So here's just a quick look at the study population. So as you can see, so these are age groups from 18 through 89. And you see that we have many more women in each age group. And also the tendency is, so we have many users from the younger population. Okay, so very quickly, the main idea, the main goal of this part is to try to estimate the prevalence of symptomatic COVID in the population currently. So I'll describe a penalized logistic regression model, which was developed by Ulf Hammar from Uppsala University to predict symptomatic COVID. So we start with 22 potential symptoms. There are several other symptoms, but this is the list we start with. We also include sex as well as interactions with all covariates, with loss of smell. So in total, we have 55 potential covariates. The final, and the model is fit to the subset of the data that is provided by individuals who have had a PCR COVID-19 test and have completed an assessment within the last 7 days. So the estimation, so I just wanted to show, so this is what the model looks like. So this part is the standard logistic model. Theta I is the probability of having an event, which is being positive for COVID. Here's our intercept and estimates and covariates that go into our model. And here we have a lasso penalty term. So we don't want to estimate all the 55 covariates. We want to figure out which ones are the most important. And here on the bottom, you see the result of this fitting. So here are the estimates, the beta 1, beta through 14. So we end up with 14 covariates. And here they are. So the most important is the loss of smell, obviously. A usual muscle pain, fever, and sore throat is negatively associated. So you see the higher the bar, that's the positive association with COVID. And the green ones below the line are the ones that are negatively associated with COVID. So then we go on to prediction. And the prediction is based on participants who have completed at least one assessment in the last 7 days. The prevalence estimate for like a given region, for example, is the mean of the individual predicted probabilities, accounting for sampling biases in age. We dichotomize age currently, so we stratify age at 50. But I guess if we had more data, we could make it finer. But at this time, we used just the dichotomized age and sex strata, and we use inverse probability weighting to account for the biases as you saw in the sample. Also, we, for example, to ensure the accuracy of the estimates, if we see that a certain region doesn't have enough contributions, enough assessments, we do not provide an estimate for that region. So this is just a brief intro to inverse probability weighting. I just wanted to for people who haven't heard of this, I just wanted to introduce it. That the idea is the idea for it is quite old, but it's still a very active area of research with a wide range of applications, and it's extremely useful. So I encourage you to look into it if you're dealing with biased samples. So here now I'm going to go into the results. So this is a dashboard that was implemented by Hugo Fittibaldi, who is a PhD student at LUDC. So the dashboard can be found here, and I think Neli will have a link at the end of her slides as well. So this is updated daily. So we provide an estimate. So currently, so as of yesterday, the estimated prevalence of symptomatic COVID was 0.43% nationwide. So here you can, so if you go to symptoms on the dashboard, you can look at symptoms that are positively associated with COVID. So and their trend over time. So we start from May, and we show you how many people have reported having such a symptom over time. And as you can see, the loss of smell is quite high since November. So in November, and Neli will go into that, so maybe I will not get into it. But so we had loss of smell, and now we have loss of smell and altered smell, a combination of these two, which is a more accurate assessment of the symptom. Okay, and here's a national. This is a national estimate over time. So the blue line and the confidence interval shows the prevalence of estimated prevalence of symptomatic COVID over time. And the red line shows the number of assessments that was used to generate this estimate. So you can see, for example, this peak here in the prevalence is due to, we think it's due to kids going back to school and just disease spreading more. Here's winter. The little peaks in the assessments would be, for example, if we had some news event and more people joined. So it's quite interesting to look at all this data. And you can also look at a map. So this is the general map of Sweden with prevalence reported for each county. And you can go deeper. You can look at you can look at 2-digit postal codes as well if you're interested. And I just want to give a shout out to the Crush COVID in Uppsala. This is an effort led by Tuve Fall to kind of identify and crush COVID and as it happens. So they keep track of outbreaks and provide testing facilities and so on to the areas, the hotspots. So please check that out. Okay, so if you take anything away from this talk, I want you to just remember that understanding how your data was sampled is key to making your results analysis accurate and results generalizable and reproducible. And I think one of the best methods one of the most flexible and best flexible, widely applicable methods for that kind of thing is inverse probability weighting. So yeah, have fun with your data. And Neli, please take it away.
I will try to share my screen fast. Oops. Okay. Does it show up okay right now?
Yes.
Okay, perfect. So I'll also briefly talk a little bit, I'm sorry. I'll briefly talk about my journey. I am originally from Tbilisi, Georgia, where I finished my high school and left friends and family to move on to Boston, Massachusetts, where I did my bachelor's degree. I did a bachelor's degree in cognitive and brain sciences, and that's also where I found for the first time that there's such a thing as programming and computer science and statistics. And it's also where people consistently kept thinking that I am from the state of Georgia. And then I moved to Sweden for a master's in public health and epidemiology, where I continued learning about statistics and just spending evenings of my own time to learn R and Python and different data analytics tools. And that led to an internship in data analytics, which led to a job at Lund University Diabetes Center, which led to the COVID project and thus this conference. So very briefly, which aspects of data science I'll talk about is the free text analysis, which we did, web scraping, and Google Distance Matrix API. So very simplified free text analysis steps that we had to implement is that I'll break it down, but first I want to say that whenever there's a package in any language, it's most of the time adapted for English. So there's a lot of things to think about when you're trying to analyze free text data. And so first of all is that we just need to make sure we filter it correctly. We do not want to include, for example, if we're comparing positive or negative test results and like their attributes, we do not want to include antibody tests or tests that were failed or are still waiting for the results. And then we also noticed that there's a lot of, probably from autocorrect, there's a lot of typos like swan and stab instead of swab. So we just want to make sure that we capture as much information as we can. The next step is case sensitivity. So again, to capture everything, it's easier to, for example, lowercase or uppercase everything. Then fishing for missing rows, that could be explicit missing values or implicit missing values, which we had to figure out. Removing all types of punctuation, as well as symbols like underscores, parentheses, dollar signs. People would sometimes mention emails, as well as numbers. A lot of people would write something for example, they had symptom X for 4 days. So we had to make sure that we removed those things as well, not to pollute our data. Lots of consecutive spaces also turned out to be a pretty frequent pattern. Then also one of the most important ones, I think, is stemming. So for example, if somebody wrote that they sneeze or sneezed or sneezing, if we do not stem, then we would have 3 different words respectively. However, if we stem, they all share the same root, sneeze. So we need to make sure that we stem correctly to capture enough information. Then, of course, removing duplicate entries and making sure that each individual can contribute to a word only once. Because, for example, if somebody decides to write a comment for 7 days in a row and they mention the same symptom, we do not want to overcount it. Then just a screenshot pretty much of some of the words we had to remove. So there is 114 official Swedish top words, but because you always need to customize, we ended up removing more than 200 words. And I think an interesting one is just this O, which it showed a lot in the plots, and then we figured it's just an abbreviation for the non-Swedish speakers for "and" in Swedish. So we also have to remove small things like that. And this is again a simplified plot of unigrams, which means that it's only looking at one word. And as you can see, for example, a lot of this big difference is coming from negative and positive, most likely because people with negative test just had a usual cold symptom. But then you also see things like high what? I'll talk about this later, or pain related to what? How can we complement this information? And so, first of all, we think it's important to try to present the same information in different ways, depending on the audience. And this is just pretty much the same plot I showed on the previous slide, but in a word cloud. And as you can see, for example, there's luktsinn, which is sense of smell. That's how we found out that, for example, people who tested positive tend to lose their sense of smell. And this ties back to this being adaptive, as Marlena said, because, for example, some of the questions being adapted or added or altered in response to the results that were found in the free text analysis. But I mentioned that some of the words that showed in unigrams don't really say a lot. So for example, high, what is high, what? Is it blood pressure? Is it pulse? And which leads to the next step. You can complement unigrams with bigrams, trigrams, however you want, but I just chose to show a snippet of some of the information you could gain from bigrams. And for example, this is a directed graph. So we found that most of the time when we saw the word high, it was associated with pulse, as well as usual, like usual cold was common in people with negative symptoms and different types of pain, headache, sore throat, back pain. And we also, I think we have a very unique set of data, but it's also very good to complement it with other data, which could be publicly available, which is why we did web scraping, politely, of course. And so, for example, in order to make sure when people provide their code, we used publicly available databases to make sure that each code was valid and that it was assigned correctly to the correct administrative region. We also used information from Post Number Service, Swedish Public Health Agency. And like we said, we think it's important to take others' information, but as Marlena mentioned, we also think that it's important to share our information, which is why you can download aggregated data from our dashboard. And it's different. You can choose what kind of level you want, but you can look more into it. And also, I think it's important to briefly talk about thinking outside the box, and again, using data or tools available. So one of the questions that came up was, is there a difference between testing depending on how far a person lives from the testing center? Which gave rise to this idea to just use Google Distance Matrix API to calculate each person's location. We only have the postal code, so the center of the postal code to a test center, and see if it makes a difference. And it resulted in a paper, which also leads me to the next fun fact, almost, that It's been almost a year since we began, and the study really believes in giving back and publishable research because we do depend on people's contribution, and we think it's really important to give back. So there have been quite a lot of papers coming out, and more are coming. And just key takeaways is that it's more of an encouraging thing rather than being very technical, but data science, a lot of it is about critical thinking and just curiosity and creativity. Don't be afraid of applying things as you learn them. Just embrace this concept of learning by doing. I don't think you need to finish 10 different courses or finish reading that textbook that you've been trying to go through before you start applying. So just go for it. There is a package for almost everything. And as you can see, we really have not reinvented the wheel in a lot of things. We just went and explored and used what people have done before and built on top of that. And especially in the context of our study, where we really depend on volunteer-based data, for us, it's really important to give back. So give back with publications, blogs, news articles, just share how the people's data is used and how they have helped you to make insights. And last but not least, just enjoy the process. And thank you.
Thank you so much for this interesting talk. I love that you're giving like a completely different perspective on this field. Super interesting. There have been some questions, but I think Marlena has already answered a lot of them in writing.
Sorry, I thought, okay, yes, I was answering them as
No problem.
Sorry, we can elaborate on some of them, yeah.
No worries, we're happy you're answering them, it's great. How about we talk about, what about you pick your favorites, and we can talk about one of those. Which one did you find most interesting, Marlena?
Of the questions, you mean?
Yes.
Okay, so somebody asked about the visualization tools. Do we use any external visualization tools? So as far as I know, we use ggplot and Plotly. I don't know if Neli, maybe you can add something to that. So
right, ggplot and Plotly.
Yeah, so we kind of try to talk about the tools we use so that people can try them out as well. Yeah, so we somebody asked about whether we plan to analyze long COVID, prevalence of long COVID. So we're looking into not maybe necessarily prevalence, but risk factors for long COVID. And so for example, comorbidities and things like that. So we're especially looking at, since we're at the diabetes center, we're looking at diabetes, whether diabetes is associated with long COVID and so on. So keep an eye out. We will be publishing something hopefully soon.
Interesting. There's also a question around sort of modeling techniques, if you've used more sophisticated methods.
Oh, yeah.
Regression. Do you want to elaborate a little bit on your answer to that one?
So what did I say? I said that we have a machine learning guy, Nikolai. So he tried various other tools, and I'm blanking on like what exactly maybe Neli will remember, but he's.
I believe, don't quote me on that, but he has tried dynamic time warping as well as different, just forecasting. But we usually share a lot of things during our team meetings, and there has been a lot of different models tried out. But we need to weigh kind of the trade-off between making it interpretable and understandable and easy to share by research and also to modify as we go versus for now it's just the model that Marlena shared.
Yeah, and so we kind of try to look at how our predictions, prevalence predictions, how they compare to the data that's available from other sources, and try to get a sense of what is the truth, right? So far, I think the best model that we have is the one that I showed today. Yeah.